Before we dive into the presentation, we'd like to take a moment to acknowledge that this session has been made possible through an unrestricted educational grant from Nestle Nutrition. We appreciate their support and encourage you to join us in thanking them for bringing us together today. Our session today will look at the early development of the human microbiota, how breastfeeding and early solid food feeding supports healthy babies. We'll explore the connection between the microbiota and developing immune systems and how probiotics impact the microbiota and immunity. This presentation will offer you an overview of how probiotics can benefit infants and will provide you with reliable resources you can turn to when recommending probiotics. I'd like to introduce you to our first of two speakers, Dragana Skokovic Sunjic. Dragana is a respected global leader in knowledge mobilization for probiotics. Dedicated to translating scientific research in this field, Dragana has authored annual editions of the Clinical Guide to Probiotic Products for 12 years. A clinical pharmacist practicing in Hamilton, Dragana is recognized internationally as a much sought-after speaker and is known for her passion, determination, and expertise. Now that you know a little bit about our expert, let's get started. Welcome, Dragana.
Thank you for introduction, Catherine, and I'm so glad to be here to speak with a fairly large group of very, I guess, curious healthcare professionals. So with the next slide, we will be talking and starting with the early development of the human microbiota. What's interesting is that with terminology, microbiota, microbiome, probiotics, connections with the overall health, there is an increased curiosity about it. I would say almost acceleration about potential for the impact on health by supporting a good gut microbiota. What we sometimes do not really realize and do not think about is where does it actually start at, and what are the factors affecting the early development of human microbiota, and if needed, how can we impact and influence that? So the question is always, where do these bugs come from? As per definition of a microbiome, or specifically gut microbiota, it's the mixture of good bacteria, fungi, and even viral community phages that live together and live well together and support our overall health. What some of the factors that we have believed actually, or we didn't know before, we believe that intestines of an infant are sterile at birth, and then within hours after birth, they are getting colonized or populated by hopefully friendly bacteria. We also know that the development of this diverse, unique fingerprint or blend of good bacteria in a gut happens in the first 3 to 5 years. The first 1,000 days in infant life are crucial for the rest of the life. Some of the researchers call this unique fingerprint a zoo, microbial zoo of the infant gut microbiota. So looking more into these factors, what we're learning lately is there is a potential exposure to maternal microbiome, even in utero. You know, as I said, traditionally the development of gut microbiota has been considered to start at the actual birth with exposure to maternal microbiota, vaginal and skin. But recent studies have indicated that there is acquisition of the gut microbiota that can begin as early as in utero by exposure to the microbial communities or communication in the placenta and amniotic fluid. This is still very early research. There's still lots of controversy, but there is definitely signaling that the communication starts much before actual birth. Its mode of delivery, vaginal birth versus cesarean section or C-section, makes a big difference. Because with the vaginal birth, we do have exposure to maternal, vaginal, intestinal, and skin microbiota. that does seem to have beneficial impact on infant development, early development of gut diverse microbiota. Going forward, feeding practices, breast milk itself provides not only bacteria, but also specific food for bacteria or prebiotics or oligosaccharides that will encourage colonization of the gut by beneficial bacteria. Joanne Hackner will talk more about that in the second part of the presentation. Another factor that is very crucial is exposure to the environment, the early stages, the caregivers, the grandmas, kisses, etc. So everything that the baby is exposed in early life. And this is something that we will be actually probably finding more about right now in the pandemic times, when we are minimizing exposure to the environment and caregivers and population surrounding the newborn babies. Next slide. Lots of research has been looking at how much impact is actually mode of delivery having on gut microbiota, and again, what is it that the babies that are born by C-section are missing. And what's interesting that, some of the factors beyond only methods of delivery and early exposure to vaginal microbiota that will actually contribute to the differences is that mothers that have C-section usually will receive antibiotics before birth, and those can cross the placenta. And babies that are born by C-section usually spend longer time in hospital and usually receive those the microbe-filled breast milk much later than infants born vaginally. So another study, you can go next slide. The next slide is another study that really looked at very specifically into the composition of the gut microbiota, comparing the first 2 weeks of the infants born with vaginal delivery versus infants born by C-section. And this study compared the cohort of infants delivered that received the same postnatal care, and again, looking at the microbiota. So what they found, they actually confirmed the previous reports that shown that delivery mode is the major determinant of this neonatal intestinal microbiome establishment. And in addition, the investigators in the study identified what is kind of how depleted the bacterial composition in the early gut microbiome of the C-section delivered infants are, and what the difference is with the vaginal delivery infants. And what's interesting that these microbes that are actually missing in a C-section born infants are those that could play a very important role in a postnatal immune and metabolic system development. With the next slide. With any time we identify something that will have impact on health, we try to figure out what can we do to fix it. So if there is a good bacteria in the gut that is missing, how about we add that good bacteria and fix it? We always, we tend to do and look for the very simplistic solutions quite often. But again, looking in probiotics and trying to understand probiotics and their impact on general health, specifically in infant health in early life, and we have this window of opportunity to impact the health for the rest of the life, it's very important. There's lots of research going on there. Next slide. Probiotics, by definitions, are live microorganisms that, when given in adequate amounts, can support health of the host of our cells. So the keywords here are live microorganisms and adequate amounts. Definition has been coined initially in 2000 and then updated a few years ago. And again, there's still ongoing discussion, how do we define and how we differentiate probiotics as an intervention versus live bacteria that are present in fermented foods that we ingest daily. Next slide. One of the facts that nobody can argue is that probiotics are popular. And again, there are some of the data that we have about expected growth, and it's in billions of dollars in the US, for example. In kids, it's the third most used natural product. In my practices and primary care in Hamilton, since I started asking my patients if they are taking probiotics, I would say 99% of them are, and most of them have no idea why. and most of them do not know what they actually take. The next slide. It is also encouraging to see that research is also following the increase in popularity of probiotics. There's more and more research being published. This is an example of a very simple query in a PubMed looking at probiotics in children, infant, baby, and looking, how many studies we have looking at the probiotics in a pediatric population. Next slide. And with the next slide. So looking very specifically for the benefits of probiotics in infants. So again, looking at the babies, they are newborn babies, they're just developing their gut microbiota. They are in position that we can have an impact, but again, we have to be extremely careful what impact we actually do on that very sensitive early gut microbiota. So the next slide. Some of the commonly used probiotic species that have been tested and studied in early life are from the genus of Bifidobacteria and genus of Lactobacilli. And here you have some of the examples of these species that have been used, and strains that have been highlighted here are those strains that have been actually added even to infant formulas. So B. lactis or Bifidobacterium lactis, BB12, Lactobacillus rhamnosus GG, Lactobacillus reuteri, DSM 17938. When we talk about the impact of the probiotics on infants and especially those born by C-section, looking at a specific study that was done with the objective to assess the effect of Bifidobacterium lactis BB12 supplementation in infants during the first 2 months or first months of life. And they was the breastfed infants that were delivered by the regular vaginal birth or C-section. And again, there was a test group infants that was assigned, they received breast milk and formula supplemented with B. lactis probiotics, and the control group received the breast milk and formula without probiotics. So the investigators looked not only at the presence and the amounts of the bacteria in stool, but they also assessed the serum immune markers in these babies and stool immunoglobulin or IgA as well. And what they have found that B. lactis is detected in significantly higher in the probiotic supplemented group, even for those with a C-section delivery. And the study concluded that the early Bifidobacteria supplementation in these infants, specifically those delivered by C-section, can be associated with the higher levels of stool Bifidobacteria. And in addition, the anthropometric data suggested that there is a beneficial effect of bacteria supplementation in this case. The next slide. So I mentioned Bifidobacterium lactis BB12, a specific strain. That's a very interesting strain that is actually one that was identified and isolated from a mother's breast milk. And again, it's one that was found predominantly in the gut of breastfed infants. And there's a lot of clinical research with a specific strain in pediatric population that is signaling some of the great support for developing microbiota and developing immune system, and also establishing good gut barrier function to protect the baby for the rest of their life. The next slide. When I was talking about probiotics and mentioning strains, there's a combination of known terminology like Bifidobacterium lactis, maybe not so much, but the series of letters and numbers at the end, it can be pretty confusing. So do we really need to know the strain and what is the name? Do we really, how important the strain specificity is? To illustrate that, I really like to use another example of a strain. You can go to next slide. Which most of us do know. It's Escherichia coli or E. coli. And there's a strain called Nisli 1917 that is actually sold and marketed globally as a probiotic supplement that has demonstrated great benefits in inflammatory bowel disease, and it's prescribed by gastroenterologists worldwide. Very safe and very beneficial. Second strain is an example is E. coli. H1SE15, which is fairly harmless commensal strain of E. coli that most of us actually carry throughout their life without any problems. And the last example is E. coli with the strain H7O157, which is extremely harmful strain of E. coli that can actually cause enterohemorrhagic colitis. And it's the strain that got into the water supply of a small town of Walkerton in Ontario 20 years ago, and the whole town got sick. Hundreds of people got in hospital and some people died. So strain specificity could actually mean difference between life and death. Thankfully and hopefully we do not get those harmful strains on our market as the supplements, but again, strain specificity effect is different for different strains. So when looking at probiotics and looking at the research behind them and what are the resources for us to pick or to recommend a proper probiotic, those are the questions that we try to answer as well with the next slide. And the reason why we need to know about them is that physicians, pharmacists, dietitians are noted as the top 5 influencers when it comes to recommending probiotics, or where the consumer or patient is actually seeking the information. So I think I'm glad that we see some of the healthcare professionals highly ranked, a little bit above Dr. Google. So the next slide. In Canada, we have the position statement that is done by Canadian Pediatric Society and was updated last year, which does tell us, in the next slide, that the effect of probiotic is a strain and disease specific. They also talk about looking at the what are the conditions that we should be recommending probiotics to. So the next, and next, and next. So looking at the probiotics such as diarrhea, etc. But with the next slide, we see there is a bit of a question that the position statement actually leaves us with. What if we need to look at the strain specification? What is the strain that we should use for what indication, and in which dose, and how long, and with what format? So those are questions that we kind of are left with missing. Last year, there was a cross-sectional survey to ask the healthcare professionals about their understanding and attitude toward probiotic. And it's interesting to see that healthcare professionals recognize that probiotics have a place and they would be a great option for intervention if we had more information to support our decision-making when talking with the patient. So this is the reason why in 2008, myself and a group of great clinicians and researchers started this project of the Clinical Guide to Probiotic Products, basically looking at the published research, published evidence with the clinically relevant outcomes, and look at the matching strains or strain combinations, and assigning it to actually find what commercial product contains a specific strain. So we made it into chart format, so make it easy for clinicians to look at what is the evidence and to sort through this humongous and vast offerings on the market. So the next slide. This is an example of one of the pages in the clinical guide. So when you just press enter, you'll see that in the first column, you have the brand names that are listed in alphabetical order. With the next click, you'll see the probiotic strains that are present in each of the products or combination of strains. Then there is, if you go across, you see what these specific strains would be used for. And with the next click, you'll see there's another one on top upper right corner. You see there's a list of indications or list of the reasons for use. Let's example, common infectious disease or cold and flu prevention. And you can go down and you see there's level of evidence, and you see in enough brackets, it's the references used for this particular strain to be assigned that level of evidence for this particular indication. So it's a very comprehensive, sometimes maybe too comprehensive resource, but again, something, I guess, to start looking at what is available on the market and what and how to recommend. With the next slide. the chart or probiotic guide, I just saw one of the questions popped up, and I'm going to answer it right now. There is at the beginning of the guide, there is information to explain those acronyms. So HP, it's H. pylori, and ID is infectious diarrhea. So again, there is a connection, you can actually see it. What I showed you, it's a printed copy of the clinical guide, and there is a Canadian and US guide. There is also a probioticchart.ca website and a similar one for the US that contains exactly the same data, maybe a bit easier to navigate because it's an interactive. You can click on a product and you can see another window will open with all the indications without needing to go with looking back and forth with looking for the abbreviation. The same information is available as a mobile app. So again, also interactive and contains all the information, including all the references. So on all three platforms, same information is replicated. So we do update the guide every year. So it's coming, next one is coming up first week of January, and we're very excited about it. So in my conclusion, for my part of the talk, I like to tell you that and I hope you agree that specific probiotics may promote digestive health, and they can really support and balance the microbiota, specifically the developing microbiota in early life. And they can also affect developing immune system in a beneficial way and improve gut barrier function. Some of the other common uses of probiotics that have been well researched in infants are the relief of excessive colic and related crying, reduction of the spit up frequency, promotion of regular stools. So there are definitely great therapeutic areas in pediatric health that probiotics can be used safely and effectively. So with the next slide, I like to pass the microphone to Joanne Hackner. Joanne is, and I'm so glad that we have Joanne with us today. She is world-renowned nutritionist, has over 30 years of experience, and primarily at the Stanford University Medical Center, where she focused on pediatric gastroenterology and medical student education. And to share her knowledge, she wrote a Gut Insight, Probiotics and Prebiotics for Digestive Health and Well-Being. There's a book, and there's also Gut Insight website where you can access lots of interesting data when it comes to prebiotics specifically. And again, this book focuses on food sources of probiotics and prebiotics, and it's based on science. And Joanne's teaching and writing conveys the belief that gut health improves overall health. So with that, I will let Joanne take over.
Thank you. Good afternoon. And greetings from San Francisco. So I'd like to continue on from the discussion of probiotics, introducing and discussing prebiotics, focusing on the first 1,000 days of life, the time spanning roughly between conception and one's second birthday, is a unique period of opportunity when the foundations of optimum health, growth, and neurodevelopment across the lifespan are established. And I read that to you because I want you to know the important role you have. Caring for pregnant women, caring for infants, advising them, consulting with them. It is a unique responsibility. And as was presented, the mother with a vaginal delivery can give a very special gift to her child, her microbiome. She can cover that baby with her microbes. So you begin by discussing it in pregnancy and then carry it on through. Next slide, please. Your body is microbial throughout. And I think it's okay to talk about microbes. I think it's fine. People are reading more about it, and you can discuss that with your clients. And the highly colonized areas certainly include the urogenital tract, where the vaginal tract is included. The low level of colonization has been thought to be the brain, the breast tissue, the fetus, and the placenta, but there is so much research going on now. And as mentioned, there is evidence of the placenta and the amniotic fluid carrying some of the microbiota. So this slide is from Gregor Reed, and Gregor Reed is Canadian. He presented this at Food Fluence. And I thought the question of, can we manipulate our own biology via the gut microbiota, the regulator of human biology, with this beautiful illustration of what we're really talking about today. The next slide. Exciting is that we can manipulate. This is a quote from the Sonnenbergs, who are microbiologists, researchers at Stanford, and authors of The Good Gut. Embedded in the numerous revelations about how the microbiota controls aspects of our health, there is a central theme that reveals how we can exert control over these microbes. How we can exert control over these microbes. The microbiota is directly responsive to diet. So this is really a cartoon-like illustration taken from one of Sonnenberg's papers, but it is such a good illustration that you can use with students and clients in teaching what really happens. So let's look at the first scenario, where the broccoli or the vegetable is going to travel through the small intestine. And you can see that nothing happens. In other words, it just passes on through, it's not digested, but it goes on into the colon, and you can see the activity of the microbes. Microbes are very active, fermenting away. And the end result are those short chain fatty acids, so important. Whereas the second scenario, the cake is coming in and is totally digested and going into circulation. So the poor microbes have nothing to do. There is no fermentation. There are no short chain fatty acids. The Sonnenbergs feel that that's contributing to Western disease. Next slide. So gut health responsive to diet, as I go through the presentation, I hope to give you messages that you can really use with your clients and your patients, in that you can influence your own gut health by means of the diet and the foods you eat. And the health of the gut is nurtured with a diet rich in prebiotics. Gut health improves overall health. So the prebiotics, giving you some history so you have a little bit more understanding about where the name came from. Glenn Gibson and Marcel Roberford introduced the concept of prebiotics and coined the word prebiotic in 1995. it was initiated to build on the probiotic concept. So we have prebiotics, we have probiotics, and yes, there is still some confusion, even among professionals. Later, Gibson, known as the father of prebiotics, developed criteria for its classification. And here you can see a young Glenn Gibson in his lab in Reading, England, with his artificial gut. So he could actually show prebiotic activity from substances. Next slide. So the criteria that he developed, it is resistant to gastric acidity. In other words, it's just like the broccoli you saw. It passes through the small intestine, basically undigested, going on to the large intestine where fermentation takes place by the microflora. Selective stimulation is part of the criteria, and I will tell you more about that later, but it is the growth and/or activity of intestinal bacteria associated with health and well-being. Next slide. So the expert consensus document, 2017, it was published, and you can see the first author is the father of prebiotics, Glenn Gibson. And then as we go on down, you may recognize some names like Mary Ellen Sanders and Gregor Reed. So they brought the experts together, and the experts developed the new definition. Next slide. It was modified. It was modified to a substrate. A substrate is something you can act upon, right? A substrate that is selectively utilized by host microorganisms, conferring a health benefit. And they expanded the concept to possibly include non-carbohydrate substances and sites beyond the GI tract. Next slide. So here's their chart. And the chart shows not prebiotic on one side, and that's where your probiotics are, and prebiotic on the other side. All of these substances affect the microbiome. Important is that selective utilization by host microorganisms. Next slide. Selective utilization was important to Mary Ellen Sanders, microbiologist. And the reason because selective utilization is a requirement for a prebiotic. Prebiotics encourage the activities of a subset of your microbiota that have beneficial functions, including those commonly known as probiotics, Lactobacillus and Bifidobacterium. Fibers may be selectively utilized as well, but this is not a requirement for fiber. It's a requirement for prebiotics. Next slide. So back to our first 1,000 days, early life factors. We've discussed the mode of delivery and the gift that the mother can give to her newborn through vaginal delivery. So now let's discuss early feeding. Advantages of breastfeeding include the mother-infant bonding, immune protection, prevention of acute and chronic diseases, and the mother's health. It's so important that the mother understand why we recommend breastfeeding. It's her choice, but let's educate her on breastfeeding. Next slide. Going back to the consensus statement and definition, I have circled human milk oligosaccharides just below prebiotics. What are human milk oligosaccharides? Next slide. Breast milk contains them. They're made in the mammary glands of the mother. Genes of the mother influence the selection. About 200 have been identified so far, and there may be more. These HMOs pass undigested to the baby's large intestine, where they are fermented by colonic bacteria. And there was one study that showed very, very early that the baby who is breastfeeding, of course, first gets the colostrum, which is very immune protective, and then gets that first breast milk, that they could find some of the HMOs in the stool of that baby. But as the baby developed and the microbiota became more established in the baby's large intestine, they did not see them in the stool. So they were being utilized in the large intestine. So the major functions are the growth of milk-oriented microbiota, dominated often by bifidobacteria. They have an important role in that they compete for bacterial binding on that epithelial, and they prevent intestinal pathogen adhesion. There's one study I recently read where the HMOs taken up the space on the epithelial and the E. coli had no place to attach. They improve host defense by modulating immunity and promoting a strong intestinal barrier function. Next slide. So the important thing, the important communication is that HMOs are protective and that we find a greater number of bifidobacterium, which promote postnatal maturation of the immune system and thus have a protective effect against infections and allergies. And that was written in 2015 by Vanderplas. So we reviewed how to establish the individual's microbiota, the newborn's microbiota. How do we preserve that pattern? Studies have shown that infant diets, toddler diets, and preschool diets influence the microbiota diversity. So the goal then is to feed prebiotic foods to enhance a healthy and diverse microbiota. Another message we give to the mother, and we can suggest the prebiotic foods, the single grain cereals, the applesauce, the mashed banana, all of these prebiotic foods, starting their feedings usually around 6 months. Next slide. Going on with banana and berries and cooked peeled apple chunks. Vegetables may not be the most appropriate at this stage unless they're really cooked and softened. But the next stage, in the next slide, you can see the baby taking the food in her hand and eating it herself. So definitely you can start in with the vegetables that are cooked and soft. Banana is such an interesting food in that it seems to be worldwide, in that when you travel, you see all kinds of bananas in different places. Somehow mothers knew that their babies tolerated them well. Next slide. So I wanted to introduce you to something that we do at Hattner Nutrition, and that is our interest in a prebiotic food source list. It's a research working document. My associate, Susan Anders, is a medical librarian, and she works on this project in actually showing a prebiotic food that's listed on our list has science behind it. And here's an example of a food she was recently interested in, agave. And you can see the reference that she has and whether or not it's a human study, which of course is what we want, and ideally that we've got two, but they're not all human studies that are on this list, but we do tell you what kind of study. And then the book details, should we write about agave, what we would want to say. Next slide. So this just shows you the actual article that she used on agave, and that it was looking at human subjects. This is our prebiotic food sources list. And you can see we have the fruits, the vegetables, the legumes, the grains, seeds and nuts and other foods. And why is this important? I think it's important because you see all kinds of listings of prebiotic foods. Somebody may be writing an article and list prebiotic foods, and they may well be, but do we have any science that shows that they truly have a prebiotic effect? This listing does have the science. These are very familiar and probably familiar to you as prebiotic sources. The asparagus, the artichoke, the banana, the whole grain breads, and certainly the onions and garlic, used in so many cultures worldwide. But these may not be as familiar to you. The burdock, the chicory, we're using the chicory root for the source of inulin. The yacon, the dahlia, salsify, jicama you may see in your own supermarkets, but the others you may see more in a cultural market or a restaurant. But why I show these is because you may be taking a history of what a pregnant woman's eating, and you may find out she's eating burdock. So I think it's important for you to recognize that and encourage it as a prebiotic food. When you're eating prebiotic foods, just a review of what happens. And this is a very simple diagram showing you the intestinal epithelium of the large bowel. Prebiotics are entering. The gut microbiota is there, ready to go into action, to begin fermentation. It has something to work on. And the fermentation end products, metabolites, also important. Short chain fatty acids. And those have a major role in protecting the intestinal epithelium and may go on systemically. There's a pH drop with fermentation. And why is that important? It's important because pathogens do not like the pH drop, so they pass on out. So we have both colonic and systemic health effects. And you can draw this diagram even and use it with your clients, use it with your patients. The importance of prebiotics. To begin educating the mother who is pregnant about the role of prebiotics, about introducing prebiotics into her diet, which she may be very receptive. One, because she's pregnant, and two, because someone may have her on iron and she may have some constipation, and the prebiotics will definitely help that. So you can find a way to really interest her and to begin to introduce those foods. Important because those foods may well be the foods that she chooses to feed her infant and her child. Establishing that microbiota pattern in early life may affect for a lifetime. Next slide. So you will get a copy of the references and additional readings that will be sent out to you after the program. And you can see the full reference here, as many of them didn't get the full reference on the slide. Thank you.
All right, that's great. Thank you so much, Dragana and Joanne, for that, the information that you provided. Lots to think about. So we have about 10 minutes left and we have a few questions. So I'm just going to throw them out there and we'll decide who wants to answer them for us. So the first one is, are there specific recommendations for doses of probiotics? My understanding is that the minimum probiotic dose should be 10 billion active bacteria per dose. Dragana, that sounds like a you question.
So that is I think, one of the persistent myths about the 10 billion is the cutoff amount of the CFU or colony forming units per dose. It really depends on the strains. Some strains are very potent, so even 500 million will provide a great benefit as shown in the clinical studies. Other strain combinations or specific strain combinations will be needed in much higher dose. So it really is to do with the strain specificities. You know, it's like using the small child to lift the car versus using the 5 big guys to lift the car. So it depends what you like to do. And this is, you know, it depends on the strain is how much you need. So 10 billion, it's not, it's a persistent myth, so it's not needed.
Okay. And I think in the clinical guide to probiotic products, there's one of the columns in there tells us for which particular strain what the CFU should be, correct?
That's correct, yes.
Okay. Okay, great. Joanne, this question is for you. And it is, does juicing or cooking the prebiotic foods affect its potency as a prebiotic?
Not really. You can go ahead and juice. You can go, you know, ideally, we would like to have the pulp, if there's some pulp left from the juicing. And the cooking, not that much. I mean, a few studies have said, yes, it reduces it a little bit, but the main thing is to get that food in there. And if it becomes a familiar food, for example, you're using it in juicing, you may well use it as a whole food thereafter. So the important thing is to get those prebiotic foods into the diet.
Okay, great. So Dragana, I think this one's going to be for you. Many products in the market contain dozens of species, some of which kill other species. What can we do to get these products off the market?
This is, I would say, it's the bane of my existence. It's interesting idea. People or companies will look at the research done with the strain A and strain B and strain C and decide, oh, great, let's put them all together and add few more just for good measure and maybe add more amount. And then we can market this as an even better combination. That's not true. We do have a proof that sometimes these bacteria will not work well together. They will actually compete with each other once they start getting into gut. So they'll be effectively killing each other. So unless there is evidence that specific combination works well together, it should not be used. And I would say with certainty that 80% or more of the products on the market are not proven, or I don't want to say useless, because there is still not proof. I'd like to compare this as putting together, I would say, let's say, I don't know, Wayne Gretzky, which everybody recognizes, some good basketball player, myself, to play hockey against the Maple Leafs. I think today, first of all, they'll kick me out of the team because I have no idea what to do on ice. And I'm not sure about this basketball player if he can do hockey. So really, if you are putting strain combination together, you have to know that that strain will click together, work together, and help each other. But again, most of the products available on the market do not have proof. What can be done to stop it or to educate everybody about it? That's a big question. This is what Alliance for Education on Probiotics is doing. You know, I like everybody to augment the efforts of the Alliance to educate patients, consumers, and physicians or healthcare professionals about the good science behind probiotics and to select proven ones. I think industry and government and regulatory bodies have great work cut out for them. But again, because probiotics are so popular, it's very, I would say, tempting for the companies to market unproven combinations.
Great, thank you. I think either one of you can probably comment on the next question, which is, starts with, there's lots of emphasis on breastfeeding, but not all moms can breastfeed, and they feel shamed and guilty for formula feeding. Do you have any thoughts about that?
Well, let me answer by saying, I think I made the point that the most important thing is that the mother understand why we recommend breastfeeding. And if she understands why we recommend breastfeeding, she may think a little bit more about it in that very, very special gift she can give to her newborn. Or she may think about, is it possible to express? But certainly the use of formula is acceptable and can be used. And there are formulas that, of course, are very special, and you can discuss that with her. First choice is always breastfeeding.
Okay, great.
And again, I just have to agree with Joanne. Yes, that we do. I think the mothers also understand the breastfeeding importance, and breastfeeding is also bonding with the child in different way. But there are definitely circumstances where this is not possible. And the formula shaming, which is very prevalent, is really not very helpful for anybody. And the really fed baby is healthy baby. And again, looking for formulas that maybe would resemble the composition of mother's milk the closest, maybe those with some probiotics that appear in mother's milk anyway, that is added, that would be a good option. Looking at, and discussing with healthcare professional definitely would be a good idea. So one or the other, the feed the baby and support the gut microbiota later in life is excellent. But again, if the breastfeeding cannot be done, that's nothing wrong with the formula.
Okay. And related to this question that you've just answered is another one. It says, do we need to supplement with probiotics if we are eating fermented foods?
Let me start and then Joanne can jump in. For a healthy person that is eating fermented foods or prebiotic-rich foods, does not take antibiotics regularly, etc., there's usually no need to supplement probiotics. There's no recommended daily intake of probiotic supplements or the level that we have to achieve. If everything is functioning normally, support the healthy gut with good food. In the cases that you do develop symptoms or you are exposed to antibiotics or you have IBS, etc., then you look for specific probiotics that have proof for this particular reason. Joanne, anything else to add?
No, I think you answered it well.
Okay,
there is a
great.
question about colic and crying, though. Formula fed or exclusively breastfed or both. It's for both. You can use drops for the exclusively breastfed, or you can use the formula for the formula fed infants.
Okay. Another one I think is an opinion piece here is that one of the barriers to probiotics is the high cost, which isn't covered by health insurance plans. Are there any advocacy efforts to help with the coverage of these?
So the cost, that is correct. Probiotics, as most of the over-the-counter supplements or fancy supplements are costly. The issue is sometimes if you are needing probiotics to address specific need and you need to take it for a short period of time, then the cost should not be the issue. As you mentioned, the colicky babies, they usually resolve the issue after 3 to 4 weeks of intervention. So it's not for the rest of their life. Again, as I say, it's really calculating what is the cost of intervention versus the cost of not resolving the issue. In IBS, for example, there are huge costs associated with the missing days of work, school, suffering symptoms and trying all the other interventions that will not work versus taking a specific probiotic to resolve the problem. So that's my take on cost. Joanne?
I think that's fine. Thank you.
But I wanted to just very quickly wrap up by reminding you that yet again that the updated version of the guide is going to be out early in the new year, and that you can access it as a PDF or download the probiotic chart in the US, in Canada or the US. And many of you may or may not know that AEProbio, although we've been around for a long time, we're sort of changing our direction a bit to improve communications and education about the microbiota and probiotics and all things that are related to that. So we've got a new website that's up at aeprobio.com that we would encourage you to go and take a look at. So I just wanted to thank the Alliance, on behalf of the Alliance for Education on Probiotics, to thank Joanne and Dragana for sharing their insights and expertise with us, Nestle Nutrition for supporting this event, and most of all, you for joining us. We hope that the presentation on the microbiota, probiotics, and infants has provided you with valuable information on supporting the development of healthy children with strong immune systems. We also hope that you enjoyed the session and that you'll join us again soon. This concludes our session. Thank you so much for being with us.
Thank you.